Hey Geeks, welcome to another video. So today we will be discussing about the chromatin immunoprecipitation, that is the CHIP technique. This is a part of the video series I'm making, that is techniques in biology. This playlist will help you to solve analytical type of questions asked in the part C of CSIR net as well as Geek. So if you want to fetch you 4 marks for this exam, do check out this video. So now, without much further ado, let's get right into this video. So let us discuss about the procedure or the principle involved in the CHIP technique. Now, before I tell you anything more about CHIP, we have done a video on EMSA, okay, where I have explained the electrophoretic mobility shift assay. And if you remember, EMSA helps you to basically determine if a DNA interacts with a protein, or rather I should say if there is a protein DNA interaction, right? If you haven't watched that video, I definitely recommend watching it before watching this one. If you understand that, understanding this will be quite simple. Now, if you remember what we discussed in EMSA, I told you it's an in vitro technique to determine if protein is interacting with DNA. Now, in vivo technique to determine the same is the CHIP technique, that is the chromatin immunoprecipitation reaction. So specifically, it is used to study the different histone modifications and if anything is interacting with the chromatin or not. Okay, so CHIP again is helping you to determine interaction of the chromatin or the DNA with the protein. Now let us understand the procedure first. This will make this more easier to understand. So imagine this is the DNA, or imagine this is a chromatin that you have in the cell. And we have a protein. Okay, so say this protein binds over here to this part, and here let's say the protein binds to this. The first step in the technique is carrying out fixation. Okay, and this is done by using the glutaraldehyde. So glutaraldehyde basically what it does is it cross-links the DNA and protein. Okay, it's a reversible chemical which helps to cross-link. That is, it helps to leave this structure intact. So that is the purpose of fixing. Okay, after fixing, nothing will happen to this. Okay, so you can move on to the next step. That is lysis. Now you will lyse the cells. So obviously this DNA is within the cell. So you lyse it and that's how you will get the chromatin and the DNA outside the cell. After you have lysed it, you carry out the fragmentation. Now this can be done by either using restriction enzymes for the purpose or you can use sonication, that is sound waves, which will make small pieces. So imagine this is one piece, this is one piece, this is one piece. For chromatin, you can imagine such pieces have been generated, right? Now, when you make such pieces, if you notice carefully, if a protein is bound to that particular part on the chromatin, that will not be fragmented. Why? Because this is protecting it, right? So there will not be a cut in the middle here. So you will get the protein attached to the DNA. Right? Now, after this, what you do is you add an antibody to the desired protein. What will happen when the antibody is added? Obviously, it is going to bind to this protein. And it is going to cause something called as the immunoprecipitation, which then you can observe. Understood? So as simple as that. This is the technique. So if there is the protein attached to the chromatin or interacting with the DNA, the antibody will recognize it and it will be precipitated. Also, another thing to remember is if it protein is bound, then that will not be cut. Okay, that piece will remain intact. Now, let us use this knowledge to solve a few questions. So here we have 3 different proteins. BLM1, BLM2, and BLM3, which were shown to basically be involved in DNA double-stranded breaks. They carried out a chromatin immunoprecipitation, that is a chip technique for these three proteins, and they got the following result. So looks very complicated, however, it's super simple. So this is at 0 minute before the break. That is, there has been no double-stranded break that has happened. So when there is no break, obviously the repair system will not be active. And at that particular condition, what do we see? We see that BLM1 is in this much concentration. BLM2 at 0 kb from the break, it is found to be this much, and 1 kb from the break, it is this much. Okay, and BLM3 is similar to what we see in the BLM1. Now they are looking at 30 minutes after the break. Means now obviously they carried out some experiment and that led to the fragmentation of DNA, and there was double-stranded break. Now when you see, say suppose this is where the break occurred. Okay, there was a double-stranded break occurring over here. This was the intact DNA, and then there was a break at this point. They must have got this, right? What they are observing is 0 kb from the break, means 0 kilo base pairs from the break. They are observing what is BLM1 there, BLM2 there, or BLM3 there, and after 1 kb from the break. So when we see, we observe that after 30 minutes, that is the repair system when it is active, the BLM1 concentration has suddenly increased, where at the 0 kb breakpoint, right? That means what you can understand, before the break, BLM1 was barely being synthesized, barely interacting with the DNA. But after the repair was induced, it must have gone and interacted with the DNA. And therefore, when antibody was added, it got precipitated. And you can see this thick band. This thick band denotes the interaction of the BLM1 with the DNA. Because if it was not, then you wouldn't have got a thick band. Whereas you found that BLM2 was present before, but now when the DNA damage has occurred, it is not actually interacting with the DNA. Therefore, you find baseline expression. And BLM3 initially was not interacting at all, but after the repair system got activated, it has been binding to both 0 kb from the break and 1 kb from the break. So what the results say, BLM1, 2, and 3 bind to DNA break sites. It's not true, because BLM2, as you can see, does not bind after the break. Before the break, yes. So this is incorrect. BLM1 binds to the break site. BLM3 binds to break site and beyond. True. 0 kb from the break means where the break occurred. So there your BLM1 is binding, and BLM1 is not only binding to the first site but also to 1 kb from the break. So this is correct. It says BLM2 remains bound to the DNA after the break is induced. Not true. We can see that after the break is induced, it has dissociated from it. BLM3 binds to DNA irrespective of the break. Again, not true, because before the break, you can't see expression. So that is the simple answer to this technique. There is another question from Chip, which we will solve now. If you are getting some knowledge from this video, please don't forget to subscribe and like it, share it with your friends, and let this channel grow. So now let's look at this question. A chromatin immunoprecipitation assay was performed to determine the specific transcription factor binding sites on the promoter of a gene. So you have a gene which is obviously having its own promoter, and you are doing the chip assay to determine specific transcription factor binding sites. So you want to see if there is a binding site for this transcription factor. Pull down was done using either IgG or antibody against the c-MYC. Okay, so against this transcription factor, this is the transcription factor c-MYC, and they are seeing that with either IgG or the antibodies to c-MYC, that is the anti-c-MYC, whether you're getting a band or not. Okay, a DNA containing c-MYC binding regions was used as a control for PCR, which is the input. Which of the following PCR representations of DNA is correct? Now see, it's a very simple thing to understand. What is your input? The input is the control, which is the first one on this autoradiograph. And they are saying that the input DNA, whatever the promoter was, it had the binding sites for the c-MYC, right? So DNA containing c-MYC binding regions was used as a control. So you should get a band definitely. So D is not an answer because they're saying no, you should get a band, the control. So D is not an option. So now remains option A, B, and C. So we know that the input has c-MYC binding regions, correct? Now they are asking us whether to determine specific transcription factor binding sites on the promoter of the gene. They carried it out using either IgG or antibodies to c-MYC. Now you can imagine if you have a DNA that has a promoter over here, which has a binding site for the c-MYC, then this anti-c-MYC antibodies that you add, the anti-c-MYC should bind and it should pull it down. So you should get a dark band, correct? So definitely here you're not getting any band. So this is not the option. Then there remains A and B. Now what distinction is there between these two? IgG is the same in both cases, correct? Now they are saying a chromatin immunoprecipitation assay was performed to determine specific transcription factor binding sites on the promoter. Pull down was done using IgG or antibodies. A DNA containing the c-MYC was run as control. Which one of the following PCR representation of DNA is correct? So if it has binding sites, then definitely a lot of the anti-c-MYC should come and bind, and that region should be amplified a lot, right? Because it will have that particular number of binding sites. So more number of binding sites means there will be a higher amplification. So if there is a binding site, then in respect to the input, the c-MYC concentration of the band intensity should be more, right? Because if it has different binding sites, you should have a higher intensity. And the higher intensity is observed in the option A. And therefore, it is a correct answer. Here, it shows lesser bands than the input. So that means that it doesn't have as many binding regions, which is not what they are looking for, right? So the most correct representation is the one that is shown in option A.